Hello and welcome to this section of virtual screening in drug discovery. I'll be talking about everything what you have been learning in this section of cheminformatics, like from the QSR and similarity search, the machine learning. So everything gets into this virtual drug discovery pipeline, and you can assume this is the final work you need to do using after performing machine learning or screening. So every methods of here which I've been seeing on that section of predictive modeling and docking and all those things, they get together, combined into a big pipeline, and that pipeline is known as the virtual screening. So we don't only do like particularly machine learning, but also various combination of methods to make it into a final lead molecule. So one of the challenges in drug discovery is that there is a huge vast amount of chemical space, and you can see it's like 10 to the power 20, and also a reduced number of proteins. Like the answer of the biological space is the number of proteins what we have, and we need to do like a large amount of computational screening, so as experimental screening to reduce the space and get into available compounds and the validated targets for a particular disease. Now the problem is like we have to choose the right molecule. The goal is to find a lead molecule which can be optimized to give a drug candidate in and move on into clinical trials. So either like what we do after finding the lead compound, we try to optimize it and performing several methods of chemical synthesis and try to optimize to form a successful drug. Since the chemical space is vast, so the estimates like there are like 1 billion compounds, which was suggested by Raymond et al., like having 13 heavy atoms. You can just see they have built a database of like close to 1 billion compounds, known as the GDB. Until now, we have like 65 million molecules in the Unichem, and you can perform search on this really large database. So Unichem combines every open source database together, and like they're having till now the largest structural database. It's pretty big, and also you can see the estimates for pharmaceutical collection is like 1 to 5 million compounds. So after this optimization, we come down to like 10 or 20 molecules. So there's a lot of computational works which is going on. So now also the high throughput screening also takes like 1 million compounds to get tested, and there's a small proportion from the 1 million, like 0.001% are available compounds for a particular target. And also large scale screening it's too expensive, and not all targets are also suitable for the high throughput screening. The typical schema of drug discovery looks like this: is like first is start with the target disease, then try to identify the metabolic pathways of that disease, and then you identify some of the proteins which are involved, the important proteins which are involved on that metabolic pathway, and which you need to shut down their activity. And if you have a 3D structure of that protein, well, that's very good. If not, what we do, we try to model the target protein using like sequence similarity and do some 3D modeling techniques to identify the structure of the protein, to predict the structure of the protein. And then move on, we have a very big virtual library. We do like virtual screening, and we also build some combinatorial libraries here of very large compound sets. We do some high throughput screening. Then we come into like the final screening methods. This is an experimental assay method, or if we have available database of drugs, we just also do some experimental screening of that. After this experimental screening, we try to identify the leads, and then the medicinal chemist used to optimize these leads into final compound. Like various methods of chemical synthesis, they try to modify the chemical groups and then try to optimize the compounds for uh, for drugability. The main concept of virtual screening, which you can see here. So the next slide shows you, what the virtual screening is. Suppose we have a 3D structure, and if it is known, if the 3D structure of a protein is known, we can do a structure-based screening, and which we call the talking. Or if we don't know the chemical structure, the protein structure, and what we do, we used to screen a lot of compounds, and from that, if there are some actives known against that protein, we do some similarity searching of a very large database to identify similar compounds which are active against that particular drug, to identify more compounds which might be activated and move on to lead optimization process. Other than that, if we have some actives known, we also do pharmacophore mapping, and if we have some inactives as well as actives after the high throughput screening, we can do machine learning as well. So pharmacophore modeling is ligand based. You can also do structure based pharmacophore modeling also. So this pharmacophore modeling comes under the actives, and if you have a lot of inactives or you have actives, you can build a pharmacophore and perform the searching on a large database. So here are the tricks, the basic steps. So if you have one or more actives molecule known, perform like similarity searching, or you can build a pharmacophore of the whole the active molecules and perform a search on a 3D database. So if several actives are known, try to identify common 3D pharmacophore and do 3D search. The reasonable number of active and inactive known, train a machine learning model. And 3D structure of the protein known, use protein-ligand docking method. So I'll be showing you the various methods of what we do, like similarity searching, how do we do pharmacophore modeling, and machine learning. I'll be talking about all these things, and so as the talking on this slide. So people in the pharmaceutical industry mostly perform this hybrid virtual screening. It's like together they perform ligand-based as well as structure-based methods. Uh, so at the start off from a database, they used to filter a lot of these molecules based on the rule of five, the admidization, metabolism, and digestion filters, and then check out the tox and remove just filter a lot of unwanted molecules, the reactives from this database, and then they perform 3D shape similarity of the compounds using the rocks or the FlexX programs, and then after the shape matching, they prepare a final database. They actually then perform pharmacophore based screening. They build pharmacophores and search large databases of compounds. So why they do the one of the uh, um, uh, good reasons to do pharmacophore modeling is that they can they don't retrieve similar compounds. They can retrieve very diverse sets of molecules, which means that like they can give you lot of diverse range of molecules having similar pharmacological activities. So then after that they perform some docking based screening, and they do some post processing of this like visual identification, and optimize the docking processes, and also do some solvation calculations to identify the potential lead compounds. Also, in other cases, if they have a structure along attached with the compound, they do some structure-based pharmacophore modeling, and from there they do the pharmacophore screening using these programs. So coming to the filtering the database, what we have, like, they used to clean the molecules first, like remove the isotopes, salts, and mixtures. They used to protonate, like add hydrogens and normalize the structures, and then they used to remove the duplicate molecules or some invalid structures from the database. And more on, they use defined or some other filter, like if you want to remove some unwanted molecules you don't want, they have a special pattern of molecules they want to remove. They can give that pattern, and they can filter it out. They also like remove the problematic moieties using the peens, which are like frequent hitters, like they are promiscuous, means and they hit lot of targets, and that's it's a kind of false positive in a drug discovery pipeline. They also filter like based on the physicochemical properties, and also apply protonation. After that, after all this filtering, they apply the protonation at this pH 7.4, and then move on to shape similarity search. So one of the important characteristics of drug discovery is you have to know what are the drug-like properties, what medicinal chemist and drug discovery scientist works on. So, and it is the one of the integral part of the drug discovery process. So there are like structural properties, physicochemical properties, biochemical and pharmacokinetics properties. The structural is like how the chemical is. So like about how many hydrogen bonds are present, how much what's the polar surface area is. So these are the terms which are very much related to for a chemical. So if you so all these things are optimized, all these properties, the structural properties are optimized. Lipophilicity is the whether it is more with the like more lipids, or they don't dislike lipids. It's also they check the shape and the molecular weight. So all these things properties are optimized. And coming to the physical chemical, they also need to check the solubility, how much is it soluble in water. So it's like people, if it is more soluble, then it's hard to reach the target. So they have to optimize the solubility so that it can the compound can reach the target. They also need to check the permeability so that it can permeable through the membranes, and also the chemical stability, like it shouldn't get metabolized to like very toxic substances and everything. There are also like biochemical properties like the metabolism, which is done by the cytochrome enzymes as the phase 1 and phase 2 of the clinical trials, and how much of this compounds binds to proteins and the tissues, and also checks the transport. Other than that, there is also the pharmacokinetics properties, so and the toxicity, like the clearance, how is it cleared from the intestine, what is the half life of the compound, and how much it is bioavailable, or it is making any kind of drug interactions with the other drugs. Like drug interactions sometimes are toxic, sometimes are useful, so they need to check that, and also the lethal dose for which it doesn't cause any lethal response from the system. So one of the important properties what people talk is the lead-like property or the drug-like property. So the lead-like is, and what do you mean by lead-like or drug-like? So for example, suppose you are in a virtual screening pipeline, and you have found some compounds, and those compounds are which get optimized later on by the medicinal chemist. Those are like lead-like. And what a drug-like is like, you have a big set of chemical compounds, and then which doesn't get optimized by the medicinal chemist, or doesn't need some synthesis treatment in chemical groups and everything. So those compounds are which we call the drug-like. So in lead-like, the molecular weight is like they used to select is from 200 to 350 daltons, and it might get like some 100 to 200 you can optimize something like that. The C log P is the partition coefficient, which they optimized is less than in range of 1 to 3. Single charge secondary tertiary ion is also preferred. It's important. They should exclude the functional groups and the promiscuous inhibitors, and frequent hitters uh, uh, are also, maybe the non-substrate peptides are also suitable for the lead-like compounds. The drug-like includes, we should remove the functional groups which are reactive promiscuous inhibitors, and the warheads. The molecular weight should be less than 500. C log P, I'm sorry, the G is missing. The C log P should be less than 5. The donors less than 5, the acceptors, hydrogen bond acceptor should be less than 10, the polar surface area less than 140, and also the rotatable bonds as less than 10. They have like observed if these things is more than a uh, particular these values, they have observed some different behavior of the compound. So that's why they have generalized this form of properties of the chemical compounds. So coming to the filtering of molecules using structural properties, first what we do some basic washing, like removing salts, unwanted elements, filtering out the atoms and the cations, filtering out metals from the structures. Also we remove the salts from the structures. So the allowed elements are carbon, hydrogen, oxygen, FPS, chlorine, bromine, and iodine. So you also check the proper atom types by adding hydrogen, and check if the oxygen, nitrogen, and carbon valencies are correct or not. We also check the formal charge of the molecules as well. So there are some filtering the reactive elements in the chemical compounds, which are kind of false positive. Means this they are they react with the proteins, and but they bind with the proteins, but they don't cause any kind of pharmacological activity of the protein. So these what we call the false positives. So they are the sulfonyl halides, alkyl halides, uh, uh, the uh, the halocarbonyl compounds, a lot of aldehydes and ketones also. Uh, so you can get this list and this Richten non-lead likeness in this paper. They have described a lot of interesting facts about the reactives molecules also. So also they remove the chelators or the synthesis intermediates, so which we call the warhead agents. They are the functional group which shows very high reactivity to proteins, due to which there is high attrition rate in the drug development. So they are like very this kind of chemical structures which were present, and they make the compounds very active, means reactive with the proteins, but they are not the right compounds for the drug development. And also recently, like 2010, they developed the pan-assay filters. Uh, the pan-assay inference compounds are this scaffold compounds, and did, where they are also known as kind of the frequent hitters to the proteins. So they need to get removed and from from the drug discovery pipeline, and there is a program to remove the compounds using the Payne's filter from from the database, from the dataset of molecules. Also there's a rule of thumb for which people used to follow, the hit selection and the lead optimization. So there are some of the parameters what they follow. So oral bioavailability is the rule of 5, which is popularly the Lipinski's rule of 5. So like orally it is available to the person. So what are the properties? So you should have a molecular weight less than 500, log p less than 5, hydrogen bond donors less than 5, and the acceptor should be less than 10, which was like given by Lipinski, and then when Locke also like shown on this paper. These are the programs which they use to calculate this oral bioavailability, like the ClocP and the ACD. So what happens if we violate this kind of rules? We find it decreases oral bioavailability. Other than that, so the Weber and Johnson, they also propose this kind of properties, what they show here, and also there's some toxic properties. If it is more than this or less than this, and the people they actually high chance of toxicity. And also the membrane permeability, the blood-brain barrier, and the general developability. Now we come to the similarity searching. What is it and how to do that? So chemical pharmaco it is like you have two compounds and you want to find the similarity between the two compounds. For example, these two compounds, they are chemically similar to each other. This is reflected in like they have a common subgraph. So this one till here is a common subgraph between these two compounds, and this indicates they are very highly similar. But when we do the pharmacophore based searching, these two compounds are not similar, but yet they have a similar pharmacological binding sites, like this this one this site, they have a similar pharmacological binding site. So they might be less similar, but they have same pharmacological activity because they have similar pharmacophores. So what is required for a similarity search is like you need to have a database. SQL or NoSQL doesn't matter. You can use either of them, or flat file of descriptors. For example, ChemFP is a tool which generates a flat file and then perform the search on the database. We also need a chemical cartridge to generate the chemical fingerprints or the descriptors for molecules. Example, RDK, OpenBabel, and there are many of the cheminformatics toolkits which are available. Similarity function to calculate the similarity, Jacquard, Dice, Tversky, which can be written in C, C++, or Python, and a function inside the SQL database. There is a chemical cartridge made on RDK, which I will show you, which I will show in the video, where I have worked on this chemical cartridge, and they have developed the functions to perform similarity searching on a Postgres database. So, talking about the fingerprints, the three fingerprints are like binary representation of the molecules. It's kind of the features. So, suppose for example, if this feature is present, there is a bit vector here of 1 and 0. We mark this position as 1. If it is not present, then we mark that position as 0. So we actually set a long list of chemicals here, and we compute the we try to find out the features of the chemical compounds, and then if those features which are present, we mark them as 1, or if it is not, we mark them as 0. So each bit in the bit string represents a one molecular fragment. It may be 1000, 2000, or 4000, 5000, or 10,000. It depends. More the features, more better classification you might get, or more better similarity you might get. Records. So what we do, when we do the similarity search, we used to match these two bit vectors of compounds and try to compute the similarity based on the two bit vectors of two different compounds, and compute the similarity based on Jacquard. So the Jacquard is defined as this B of x and y. B of x is the bit vector of x, and bit vector of y minus the bit vector of x and y, and on top you have this is the common bit vectors. These are the common bit vectors, and then after that we divide these two and we get the Tanimoto or the Jacquard coefficient. Or we can compute the Euclidean distance, but this is not much used. The Tanimoto is very popular. Along with that, Tversky is also very popular to compute the similarity between the two compounds. So there is a video which I did how to perform similarity searching using the Postgres database using the RDK. You might want to watch this if you want to like start your similarity searching with by yourself. Also, what we do, as I told you, in the virtual screening hybrid virtual screening pipeline, we also do 3D database similarity search, like the shape-based search, what we call the ROCs, which is rapid overlay of chemical structures. And also there is another program which is an open source, which called ShapeIt. So these are like very expensive, computationally expensive programs, and then the 2D methods, because They are the 3D methods, and they are used to compute this 3D volume and shape of the molecules and match the 3D volume of the two molecules using some integrals, and which makes it more expensive. Also, you need to generate a lot of conformers of the molecules. So in 3D space, the compounds can exist in various different conformations. Conformations means the orientations in space. So we don't know which is the right position or right orientation. It might be active in a compound. So what we do, we used to generate a lot of conformers, and while do the 3D searching, they used to take these 3D conformations along with the chemical features into account to generate a final score of the molecule between the two, and which makes them like similar. So in the 2D methods, we have bit vectors. In 3D methods, what we generate is not only the features but an extra form like the conformations for each molecule, the orientations in space, which we take into account and compute the similarity. That's why it makes this more expensive. So there is a video on rocks here, and if you want to check how to do the 3D based screening, the rocks is it's not an open source tool, but it's free for academics to do to perform this screening. It's available on Windows as well as for the Linux. It also runs on Mac computers also. So now we come, if we have suppose multiple actives known, and we do pharmacophore searching. This pharmacophore is defined by the IUPAC, is ensemble of steric electronic features that is necessary to optimize the supramolecular interactions between a specific biological target to trigger a biological response. For example, a chemical compound should have hydrogens or aromatic groups, hydrogen bond acceptors, rings, hydrogen bond donors, like the charged groups and all. I didn't show everything here, just to give you a basic idea of what pharmacophores are. So these are the kind of properties which we look for in a molecule, and the orientations in space, how they are oriented in. So it is a set of features that is so pharmacophore is a set of features which are common for a set of series of active molecules along with the position in space. So what we do, you try to align the molecules together and try to find out if the properties or the set of features are correctly orientated in 3D space, so that it is interacting with the target. So for example, the right position of the acceptors, donors, not charged groups or hydrophobic regions. So these groups are which we called the pharmacophoric groups. For example, here what we see, spatial, which shows the spatial relationship, like for example the distance, ranges and angles. We also give some special we also relax some of the distance between these positions. For example, this two, oxygen, and nitrogen, we look for these three positions here and in this molecule. See, they are not similar, but they have the same orientation of this nitrogen, oxygen and nitrogen, and the distance between this is something like this. So if we find like the distance are kind of okay between these positions, we used to collect those molecules and we generate lots. So in pharmacophore we also need to generate a lot of conformers, and conformers, and then after that we used to find the right molecule to get, like which will have the 3D pharmacophore. uh, So first, the workflow is kind of very simple. You collect a set of experimental compounds for a target. Now select a threshold of activity values. For example, if you take 1 micromolar as the threshold, so for example, the compounds above 1 micromolar should be inactive, and compounds less than 1 micromolar should be active. We create a training set from that of test and train. We generate conformers and create a pharmacophore model. So now that you can prepare a lot of pharmacophore models, but the validation of the model is you need to test a lot and perform some perform some analytics on this sensitivity, specificity and yield of actives, and the enrichment factor for the test set and how does it performs. So it's not like you build a simple machine learning model then it's done. So a lot of effort goes on to find the correct pharmacophore model. Of course the software does that for you, but you need to select a right model for the activity of the molecules and perform some statistical analysis of the models based on the enrichment score, the yield of active sensitivity and all. And then with that's your final selected for model, which based on this statistical analysis, and you perform a 3D search of the database of molecules to get some compounds, and then you do some finally you do some screening of this, like based on docking, and then you do some experimental testing of the molecules. If you want to consider more on this cheminformatics and for modeling, you might want to go to I571 Wikispaces, which is the cheminformatics course, which discusses a lot of pharmacophore modeling, and also I have made a video on how to perform ligand-based pharmacophore modeling using Ligand Scout. If you are interested, you can watch it here. These are the tools used to perform the pharmacophore searching. The Axaries has Catalyst, Schrodinger has Phase, the Ligand Scout, Intelligent has the Ligand Scout, the Pharmacist, Pharmora, Shafts. These are the open source versions of the pharmacophore modeling, but they don't come with everything like searching with database. You need to work more on this open source versions by yourself, like with statistical modeling and everything. So suppose you have many actives and inactives known. What we do machine learning methods. First, we used to build a structure activity relationship using this machine learning method. So it's a very very old method. So which was developed by Corwin Hansch, and there's also the Fee-Wilson analysis. And this QSR is restricted to a small homogeneous data sets which we require for lead optimization. So every lead optimization process has a QSR method in it, implemented in it, to find out which are the chemical backbones of the properties which are involved in the activity of the molecule. So also the SARs can be used with like data consisting of diverse structural classes and multiple binding modes. There's also some resistance to noisy data from HTS. So what we do, we also do some binary classification of the molecules and build like lot of models based on SVM or random forest and other techniques, and the resulting molecules we do some fusion of the molecules and fusion of these methods and try to prioritize the compounds for lead finding. There is also a video on this you have already seen last classes about the predictive modeling in cheminformatics which is on QSR, and also the binary classification based on different methods. So now we come to one of the very important virtual screening method is the protein-ligand docking, which we do after all this pharmaco screening, similarity searching, pharmacophore modeling, the structure and ligand based. We come down to the protein-ligand docking. In protein-ligand docking, we have a target along with the ligand which gets binds to this active or the binding site, and which causes some pharmacological activity of this from this binding. It predicts the docking predicts the pose of the molecule in the binding site, and also the binding affinity or the score representing the strength of the binding. So it represents a compound should it bind to this place or not, and also what is the score of this binding? Is it a good score or not? Or very good score represents that okay, this binding site accepts this ligand, and also it predicts the pose, like how it is oriented in geometrically inside this cavity or inside this binding site. So the docking actually predicts these two very important things. The binding site is what we call the active site, is a part of protein which where the ligand binds, or it's generally the cavity. We get actually we they identify it from the crystal structure. There also, which is available in the PDB. You can find a lot of crystal structures in PDB which has these ligands bind it to the proteins, and they have crystallized the structures. And the pose is the binding mode, is the like the location, the orientation, and in what conformation the molecule gets bind uh, to this binding or active site. So for ligand docking, how does a ligand bind to the active site of a protein? So docking algorithms are based on the two key components, the search algorithm and the scoring function. The search algorithm is to generate poses of the ligand within the active site, the conformation, position, and orientation. So generate poses like it binds, and it generates a lot of conformations of the molecules along with the orientation and the position, and try to score on each of these poses, and then optimize this lots like 1,000 poses into like the final 10 poses, which gives you the highest score. And also the scoring function on how to score the poses for the individual ligand with the target. The search space, the difficult to the protein ligand docking is due to the fact involves many degrees of freedom. It's like from translation to rotation of one molecule related to other involves there are like 6 degrees of freedom. In addition, there are also conformational degrees of freedom from the ligand and protein, and also the solvent or the environment in which the protein binds to the ligand, the plasma, is very much it plays a very significant role, and the pH of the environment plays a very significant role in the protein ligand geometry and bindings. Uh, a search algorithm generates the poses, the orientation, and the conformations of the molecule inside the binding site, and it search the space exhaustively inside that binding site. Uh, yes, there exists a trade-off between the time and the search space coverage. You can find more at this Leach and Gillett book about the search space, and how does it do does this trade-off. We'll talk about only one talk algorithm, that is the talk algorithm, but there are varieties how people are thinking right now. So doc was developed by Kuntz et al. Based on shape complementary, and also the rigid ligand is one of the very popular, very old methods of docking. But nowadays there are methods of like fragment-based methods of PlexX and doc. There's a more simulated annealing in Autodoc, and also ligand fit has that. Also the genetic algorithm based the gold and Autodoc, and systematic search which was implemented in the Fred and the Glide program. The Glide is very popular and is one of the best tools to do the docking. You can read a review of the small molecule protein docking methods on this publication. It's a very nice publication of the different methods which has been developed for doing docking. So I'll talk about the talk. Actually, the talk is a rigid docking based shape method. So actually, some spheres are constructed inside the cavity. Spheres are of very varying sizes, such that each surface of the sphere touches the 2 points of the cavity, and the center of the spheres they used to map it to the ligands. So ligand atoms which get mapped to the centers, so the distance between atoms equal the distance between the spheres. Uh, these matches are then the ligand are positioned inside the active site, and there is to check if there are any steric like bulky group clashes inside the cavity with the ligand. Now after that, they orient the molecule, they generate conformer, and they try to map it, and then again they used to score for that pose of the molecule. This way, like, they generate a lot of poses for this molecule and try to find out which of the poses gives the best results for for this molecule and active site. This is how they used to score the poses. There are some energetics which are involved for the ligand receptor binding. They are the electrostatics, including hydrogen bond interactions, the dispersions, the hydrophobic interactions, dissolutions, like surfaces buried between the protein and the ligand, the conformational changes. The ligand must be properly oriented, and also the entropy of the ligand due to being fixed in one conformation. So these are the terms. The free energy of binding involves the free energy of the solvent conformation, then interaction, rotation, the translation, and the vibrational energy. So translation and rotation and the vibrational energy. So all these terms get added and ultimately gives the binding free energy of the molecule, the binding energy of the between the target and the molecule, and which gets scored by the scoring algorithm. So conclusions of this: there are many doc programs, like there is different types. So, and there are also different scoring functions and different search algorithms, different approaches of doing docking. So if you want might check, you can check this section from Young, the computational drug design. Also gives a lot of overview of different packages. So conclusions: a wide range of virtual screening techniques have been developed. The performance varies on different data sets. The data sets is very important while you're doing virtual screening based on these different methods. So actually different methods get integrated together in a big virtual screening pipeline, and then we try to filter lots of molecules from this. We don't do we don't do a particular set of methods like only machine learning and then get those molecules in testing. So there are a lot of efforts of these different methods which come into play. So they develop like 2D, 3D, and this the docking pipeline together, and then finally they visualize they get the results. So it's not a single method. The virtual screening is a combination of all these methods together, and which helps to optimize or find the lead molecules. So now also you have learned about the computational filters. So you need to optimize the filters. Also you need to optimize the compounds based on the filters also, to remove the undesirable compounds together. I hope you understand the pipeline pretty well. So which is on this section, the hybrid virtual screening. This one, so we start with a database and then we move it to shape-based similarity. We do a lot of screening of this, the pharmacophore, docking and post-process, then into potential lead compounds, and do the uh, experimental assays, and then into final compounds for mice or mouse testing, and then into clinical trials. So these are the references you might want to see, check out for if you want to go for virtual screening and want to